Just like there are abnormalities that exist in our autosomal chromosomes, there can also be abnormalities in our sex chromosomes. And this is what we're going to focus on in this lecture. We're going to examine three different types of abnormalities on our sex chromosome. So we're going to discuss an abnormality known as Klinefelter syndrome, which is basically described by the XXY karyotype. Then we're going to briefly look at the XO karyotype, which describes Turner syndrome, and then we're going to finish off with the XYY karyotype. So in individuals with Klinefelter syndrome, there is an extra copy of the X chromosome. So instead of having, let's say, the XX or the XY chromosome, we have the XXY chromosome. Now, the reason that this exists is a result of non-disjunction that can take place either in meiosis 1 or meiosis 2, more specifically in anaphase 1 or anaphase 2 of meiosis. So to see exactly what we mean by that, let's take a look at the following diagram that describes non-disjunction taking place in anaphase 2 of meiosis. So let's suppose we have a normal female individual, and that normal female individual produces X cells. So this is the precursor cell that eventually gives rise to 4X cells. So because we're dealing with a normal female individual, we have two X chromosomes, X chromosome number 1 and X chromosome number 2. Now, during interphase, before meiosis actually takes place, each one of these X chromosomes is actually replicated. So we have this X chromosome number 1 and its identical sister chromatid, and X chromosome number 2 and its identical replicated sister chromatid. So we have a pair of these sister chromatids. So we have interphase taking place and then meiosis begins. So let's fast forward to metaphase 1 of meiosis. So this is when our tetramers basically line up at the equatorial plate of the cell. And also what happens is the mitotic spindle apparatus forms, and these spindle fibers permeate, extend from these centrioles, and they are supposed to attach, at least in a normal case, to these sister chromatid pairs. So let's suppose that this process takes place normally. And so what that means is these will segregate, will move to opposite poles, and after meiosis 1 takes place, we form these two haploid cells as shown. So haploid cell number 1 and haploid cell number 2. Now, let's assume non-disjunction takes place, and let's say that non-disjunction takes place within this cell. So what happens is this centriole basically fails to extend the fiber, and that bonding between the fiber and this particular sister chromatid fails to actually take place. Now let's suppose that non-disjunction does not take place within this particular cell. So this cell correctly undergoes meiosis 2, and so when these two identical sister chromatids segregate to opposite poles and the two cells form, these will be the identical sister chromatids, the identical cells that will contain the identical correct X cells. Now, because non-disjunction took place within this second haploid cell, because this failed to form the proper connection, this entire pair of identical sister chromatids basically moves to opposite side, and none of those sex chromosomes move to the other side. And so eventually when we form our two X cells from this particular cell, we form cell number 1 and cell number 2. And these two cells, cell number 1 and cell number 2, are abnormal. They're called aneuploid. And that's because this one lacks a copy of a sex chromosome, and this one has an extra copy, one more than it should. So we have one copy and the second copy of that, additional copy of that X sex chromosome. So notice that two of these are normal, these two are normal, and these two are abnormal. So let's suppose The normal female individual produced these abnormal gametes as a result of non-disjunction. And now let's suppose this individual mates with a normal male individual that has normal sperm cells. So if a normal sperm cell that is carrying a Y chromosome fuses with cell number 2, we're going to form a zygote that contains 3 instead of 2 sex chromosomes. And because we're dealing with this abnormal X cell, we have 2 X chromosomes, 2 identical X chromosomes. And so, because these 2 identical X chromosomes fuse with the sperm cell that contains the Y chromosome, we see that the genotype, the karyotype, will be XXY. And so this zygote will eventually form an individual, and that individual is said to have the XXY karyotype, which is also known as Klinefelter's syndrome. Now, what are some of the symptoms of individuals that have this syndrome? Well, because they have that Y chromosome, these will be males. Now, they will be unusually tall, and because of that additional X chromosome, they're going to have female-like breasts. And about 50%, about half of them, will have some type of mental disability. However, they do live normal lives. So they generally live normal lives. Now, let's move on to the X or the XO karyotype. Now, in individuals with Turner syndrome, they have one X sex chromosome and they lack the second sex chromosome. And the reason we use the O symbol is to basically represent the fact that we have an absence of the second Y chromosome. So let's see how this can actually arise. So once again, this can arise as a result of non-disjunction. But now let's study non-disjunction as it takes place in the male individual. So in this case, we began by assuming we have a normal female individual. Now we're going to assume we have a normal male individual, and that means we have one X and we have one Y. Now during interphase, we have replication taking place, and so we produce this identical sister chromatid with respect to this one, and this identical sister chromatid with respect to this one. And if we fast forward to metaphase 1 of meiosis, we basically get these chromosomes line up, the tetramer of chromosomes line up at the equatorial plate. So once again, let's assume that non-disjunction takes place in anaphase 2 and not anaphase 1. And so what that means is these mitotic spindle apparatuses or the spindle apparatus basically forms correctly, and these fibers extend onto these sister chromatid pairs. And now segregation takes place and we form these 2 cells as shown. So these 2 haploid male cells. And now once again, let's assume that non-disjunction took place during the second phase of meiosis. So we have metaphase 2. Now this cell basically divides normally, and so these fibers are able to attach to each one of these sister chromatids, so this one and this one. And so what happens is, because non-disjunction does not take place, they segregate correctly, and so eventually they will form this normal cell number 1 and normal sperm cell number 2, where each and every one of them will have the X chromosome. On the other hand, because non-disjunction, we're assuming non-disjunction takes place within this cell, once again, this spindle fiber is not able to connect correctly with this sister chromatid. And so both of these sister chromatids are connected to this spindle fiber, and they are pulled to that side during anaphase 2 of meiosis. And so what we end up forming is these 2 abnormal sperm cells. In sperm cell 4, we lack any sex chromosome, and in sperm cell 3, we basically have 2 identical Y chromosomes. Now, if we take a normal X cell that contains a single X chromosome, and that normal X cell combines with this abnormal sperm cell number 4, then what happens is the zygote that is formed will only have a single sex chromosome, the X chromosome, because there is no Y chromosome within this particular sperm cell. And this is what we mean by the XO karyotype, also known as Turner syndrome. So when a normal X cell combines with the aneuploid sperm cell number 4, the zygote will contain an XO karyotype, and that zygote will eventually develop into an individual that is said to have Turner syndrome. Now, what are some of the symptoms of this particular syndrome? Well, notice that because we don't have a Y chromosome and the Y chromosome is needed to develop that male individual, these individuals with Turner syndrome basically develop into females. However, these females are sterile, and that's because that second X sex chromosome is needed to actually fully develop the ovaries. And so these individuals are basically sterile because they do not have fully developed ovaries and fully developed female genitalia. Now, let's move on to the final type of sex chromosome abnormality, the XYY. Now, unlike the XXY and the XO, this is not actually a syndrome because individuals that have this type of karyotype live normal lives and they don't really have too many abnormalities. They have a normal phenotype and they are fully fertile. So suppose that instead of combining sperm cell number 4, we took sperm cell number 3. So we took sperm cell number 3 and we combined it with a normal X cell as shown in this diagram. What would we form then? Well, we have two identical Y chromosomes, one X chromosome, so we form a zygote that has an XYY genotype distribution, and that's what we mean by an XYY karyotype. Now, it turns out that zygotes with the XYY karyotype, the XXY genotype, the XYY genotype generally develop into fertile males. And aside from being unusually tall and having extreme acne problems, they are generally normal. That is, they are fertile and they can basically form offspring. They can basically mate with other normal female individuals to produce offspring. So these are the 3 different types of abnormalities that exist on sex chromosomes.